New-cleotide,
The Discovery of Two New Bases
by Samantha Dean
and Allison Smith. DNA makes the world turn, both literally and figuratively. Every living organism on Earth is made up of DNA, which makes the concept seem complex. When you get down to it, however, DNA is much more complex than one would think. The things it can accomplish are what make it the most important molecule ever discovered.
One of the most important components of a DNA molecule is a nucleotide. In the year 1925, a man named Phoebus Levine discovered that there were 4 nucleotides and even coined the term nucleotide itself. The 4 nucleotides that Levine discovered are called adenine, thymine, guanine, and cytosine.
The 4 nucleotide bases all pair with another in order to keep a functioning molecule of DNA. Adenine pairs with thymine and cytosine pairs with guanine. These pairs are held together with hydrogen bonds, 2 between adenine and thymine and 3 between cytosine and guanine. From the time that the nucleotides were discovered and named in 1925, there were no thoughts that any other nucleotides could exist. But this all changed in the 1980s.
This change came from the intensified research on a possible 5th nucleotide base, methylcytosine. The main function of this nucleotide base is to switch genes on and off depending on the needs of the organism the base is working in. The discovery of this base was revolutionary. Beliefs that had been upheld for the past 55 years were suddenly being altered, proving to people everywhere how complex DNA actually is. There was still more to come, however.
A few years after the discovery of methylcytosine, another possible DNA base was discovered. This 6th base is called methyladenine.
Methylcytosine was first discovered by a man named Roland Douglas Hotchkiss in 1948. Hotchkiss was able to separate the nucleic acid bases from DNA from a calf thymus. When separating these bases, Hotchkiss discovered a methylated cytosine, which was distinctly different from normal cytosine. Though it was first discovered in 1948, the in-depth research on methylcytosine started in the 1980s, and a fuller understanding of what methylcytosine actually functioned as was achieved.
Methyladenine was first thought to exist by a man named Manel Esteller in a magazine titled Cell in 2015. Esteller suggested in this article that eukaryotic cells may have this 6th base present. Though there has not been much extensive research into the existence of this base, the publishing of this article opened a door into the possibility and provided evidence that makes it very probable.
Methylcytosine and methyladenine have very similar functions. These functions include telling genomes what to do and when to do it in order to keep a functioning organism. These 2 newer bases are thought to only exist in prokaryotic cells, which are cells that lack a nucleus. However, recent research has suggested that these bases could also be present in lesser functioning eukaryotic organisms, which are organisms in which the cell contains a nucleus. These organisms include worms, flies, and some algae.
Since methyladenine and methylcytosine don't pair with anything, we think that it's possible that it's really 2 bases that are harder to separate than adenine, thymine, cytosine, and guanine.